Hello.
Hi,
Hi.
I'm here. I saw Dick yesterday.
Oh, you did?
Yes, we were talking about Tensegrity to Stephen Levin.
Oh, good. That's great. What was the outcome of that?
Well, he says my Tensegrity, what I've got, is a squish.
Oh, yeah.
In other words, the elastics on it are not like in a normal Tensegrity. There's not.
Hello? Yeah, I'm here.
Having some trouble, or?
I don't know. It just hiccuped, I guess.
Oh, okay. Well, anyway, yeah, I need to find some fishing line and fix my little toy so that it's more like a Tensegrity. They behave differently.
Oh, okay.
Yeah.
What is he calls it? A squish?
A squish, yeah.
Which is like, what does that mean?
Squish.
Like it's just not tense enough, or?
Yeah, it's not tense enough in between.
Oh, okay.
The nodes, so it's a squish rather than a Tensegrity. So that's easy to fix here.
Yeah.
This thing's elastics are going, so I need to do something with it anyways.
So what is why does it behave different? How is it supposed to behave with it?
Well, when you start pushing on it, the elastics lengthen.
Okay.
Instead of the force being transmitted through the whole structure, it lengthens the elastic instead of pushing the force along the structure.
Oh, okay.
That's what I'm seeing.
Yeah.
So that was nice because Dr. Zhang, who I'm working with, he's very intuitive about mechanics, and he said, yeah, you want it as a tension structure only. And so I'm going, yeah, it's a tension structure, no compression whatsoever, and no elasticity. Well, there is some elasticity in any material, but it just needs to be a stiffer material, which is okay. I could do that. It's easy when you're making a model. I'll just change this. Yeah,
it's great. I was reading that email that he sent to. Oh, I got the email you forwarded to me, and one of the things that was from Stephen Levin. One of the things he was saying is that there's like in a biological tensegrity structure, even the struts are like have I guess an elasticity. I guess he's talking about actin or whatever, and that needs to be accounted for. It's almost like a fractal.
Yeah, the struts would be microtubules.
Microtubules.
And he says they're like kind of like an island of compression in the sea of tension.
Oh, okay.
It's sort of a neat way to look at it.
Yeah.
But yeah, I want to know where the microtubules are now in a normal cell, a normal epithelial cell. And do you know anything about that?
Well, I mean, I don't know. Like, I don't have a good like drawing of it or an animation, but I guess the microtubules are sort of things that hold the structure together. So, like the the membrane, it holds things together. There holds the structure, in a certain conformation, and it also plays a role in some of the other structures, like the nucleus and other things that are sort of, I guess, whenever you need structure in a cell. I know they have microtubules and neurons a lot, and they do a lot of things there. Hello.
Yeah, the microtubules and the neurons certainly create the axon in it. In that. Yeah, I just asked, so where are the microtubules in a cell? Like, I know the actin in a normal non-dividing cell, etc., is on the outside like it forms an outside layer. There's stuff inside too. Yes, of course. But it does tend to the outside of the cell. At least I have a paper on that, and they, like I said, they're biologists, and they took a picture of it. This is what happened to the cells they were looking at. Sometimes it'll come into the center in odd cases, perhaps when the cell's dividing or doing something else. But normally it's in this outer coat of the cell, so at least the densest part of it.
Susan?
So we're at microtubules.
Okay. There are two sets of microtubules in axolotl epithelial cells. One set is along the long axis of the cells, perpendicular to the surface, and the other set is at random at the apical surface.
Oh, okay.
Okay? The apical surface plus the microfilament ring plus the intermediate filament ring are what constitute the cell state splitter.
Okay, there's an intermediate filament ring there too?
Yep.
Oh, okay, because I know there's certainly intermediate filaments usually around the nucleus too, but
yeah, Chris Martin found that.
Okay, yeah. So there's the microfilaments, which are actin, and then intermediate filaments.
Also like a ring. I think it was below it.
Oh, below it, there's a...
Below it, maybe a couple of tenths of a micron.
Okay. Do you have any papers on that?
Yeah, that's Chris Martin's paper.
Oh, the Chris Martin's paper. Okay,
yeah, you can read it in English or Russian.
Yeah. Oh, sure. How much below 1 micron? 1 micrometer?
I don't know. Paper. I forgot. It's been a long time.
I'll read it. The actin.
Okay.
And then.
And what was her name? Beth Burnside is the one who discovered where the microtubule, or the two sets of microtubules.
Is Burnside?
Yes, Beth Burnside.
Oh, Beth. Okay.
Yeah, she discovered them in Anton Jacobson's lab, and she wrote about them. So look up papers by Beth Burnside. I think it's under B, Burnside, instead of Elizabeth. But
oh, B Burnside, not B Burnside?
I think so. It's also with Anton. The co-author would be Anton Jacobson.
Oh, okay.
With an S-O-N at the end.
S-O-N? Yeah, all right.
Okay.
Okay, all right. Chris Martins. He didn't talk to me much when I visited him.
Oh, he didn't?
No, he didn't want to talk about science.
No. Yeah, no, he's out of it.
Yeah, he just didn't want to. And I said, well, I came all the way, basically all the way here to talk to you about this, so we can spend 5 minutes talking about this. I don't think he, like...
Yeah, I tried to rope him in again. I tried to rope him in things over the years, but no response.
Yeah, no. He got mad at something and was permanently mad about it.
Yeah, Chris Martin was a student as an undergrad, and went on. I think he did zebrafish work, and then he ended up as a professor at the University of Ottawa.
Mm-hmm.
And he got disgusted with that and moved to Granada, where he taught 500 medical students who couldn't get into medical school in the U.S.
Yes, that's what I understand. There are lots of doctors in the U.S. there that have gone to Granada and got their medical license, and then had to jump through a few hoops to get back into the U.S.
Yeah.
We did it.
Yeah. Well, I remember the University of Manitoba had a 90% rejection rate.
I've heard about it from my daughter Nancy. Like she was
and you know, and that's why I wrote those papers. That paper on the physician scientists, because one of the things they do is they complain that there aren't enough physician scientists, and then reject anybody who could possibly be a scientist to get into medical school.
So we talked a few weeks ago about something called morphological waves, and we had some skepticism in the group about the concept and how it's similar to differentiation waves. So this idea goes actually back, I think, to discussions with Christoph Tücher, who's actually a pretty interesting person in the field of A-life. And I've gathered some papers of his on chemical computation and perceptrons. So let me take a look. Let's take a look at some of these papers. So the first paper is online learning in a chemical perceptron. And of course, perceptrons are very early neural networks. They're systems where you have a lot of inputs that are summed into a single unit or neuron, and then there's an output. So you're taking in a lot of different inputs and you're producing classifications from that. And perceptrons go back to the 1960s. And they've been used in as toy models a lot for different things. In this case, we're looking at a chemical perceptron, and so we're looking at biological learning, such as it is. And so this is Christoph Tücher is the second author on this paper. He's at Portland State University. So the abstract of this paper reads, autonomous learning implemented purely by means of a synthetic chemical system has not been previously realized. So we're interested in autonomous learning in a chemical system, in a synthetic chemical system. This is a lot to do with like synthetic biology and other applications like that. Learning promotes reusability and minimizes the system design to simple input-output specification. So again, you have this perceptron where you have multiple inputs to a single unit, which either sums the inputs or filters the inputs using some learning rule and then gives you outputs. In this article, we introduce a chemical perceptron, the first full-featured implementation of a perceptron in an artificial chemistry. So artificial chemistries are actually an interesting area of A-life research, and it involves taking chemical worlds or chemistry systems and simulating them in silico. So you can simulate a chemistry system, say, to understand the origins of life or to understand how to design different types of biochemical structures, things like that. So this is a very useful thing in these artificial chemistries, to be able to learn online, to learn simultaneously with sort of exploring the space of this chemical system. Perceptron is the simplest system capable of learning, inspired by the functioning of a biological neuron. Our artificial chemistry is deterministic and discrete time, and it follows Michaelis-Menten kinetics. So this is something that's, they're trying to model this as an actual chemical system. They're using a Michaelis-Menten model. They're using a discrete time model and a deterministic model. Those are the properties of their artificial chemistry. We present two models. So these are the properties at the top, and then the second sentence they talk about their different types of perceptron that they're using for this. The weight loop perceptron and the weight race perceptron, which represent two possible strategies for a chemical implementation of linear integration and threshold. Both chemical perceptrons can be successfully identified, or can successfully identify all 14 linearly separable two-input logic functions. So these are things they throw at the perceptron to get it to classify correctly, or to test to see if it can do this, and maintain high robustness against rate constant perturbations. We suggest that DNA strand displacement could, in principle, provide an implementation substrate for our model, allowing the chemical perceptron to perform reusable, programmable, and adaptable wet biochemical computing. So one of the criticisms of the Morphological Waves was that it was sort of like it had a sort of a mysterious component to it. So there was a learning component to the reaction-diffusion equations, for example. Does that occur by magic, or is there some sort of underlying is there maybe an underlying intelligence? And even that seemed a little bit outlandish. But this is sort of addressing this issue where you have a chemical system or a biochemical system that has this learning capability. So you can actually create perceptrons in natural systems using a variety of techniques. I mean, the perceptron is just an abstraction. You can use this sort of model in chemical systems. So, for example, if you have a lot of different combinations of something that go into a process and there's a simple input-output, that would be considered a perceptron, even though it's not a perceptron or a biological neuron per se. It's that sort of structure of the problem or the structure of the system where things get processed. So this is I think this is a useful mechanism, or at least a useful model for looking at some of these chemical or biochemical mechanisms. So let's see if we can get down to the bottom here. The contributions of their model here are as follows. Our system is the first full-featured implementation of online learning in simulated artificial chemistry called the chemical perceptron. So learning as well as linear integration of weights and inputs, which are integration at that single unit that we mentioned before, is handled internally. They provide a systematic method that maps the variables of a formal two-input perceptron to species of the chemical perceptron. So there's this chemical perceptron that is based on biochemical principles. So you can go from first principles to this sort of information processing. The chemical perceptron learns perfectly all 14 linearly separable logic functions after 200 learning iterations. So it doesn't necessarily take that long to learn. The chemical perceptron is reusable since it recovers its internal ready state after each processing event. So I want to get down to this weight loop and weight race perceptrons to give an idea, because they talk about the artificial chemistry here and there are details to that, and there's a lot of kinetics in this. So the artificial chemistry it's really based on Michaelis-Menten enzyme kinetics. So if you know that, you can go through that. Representation of variables. So we have to define our variables in the computational system, the system inputs and outputs. So the concept of actions or action series is an extension of the input configuration. Chemical species concentrations can be modified at times other than T0. An AC action emulates a step in the execution of an experimental protocol, where at a certain time the person performing the chemical experiment mechanically injects or removes substances in or out of from a tank. And so this, the tank metaphor can be used in self-organizing chemistries as well, in terms of like the supply of a certain chemical reactive species or whatever. And so you have this rate limiting aspect to it that you can use to understand both sort of, if you're doing chemistry experiments or you have these self-assembling chemistries as well. And so they kind of show some of these concentration versus time step dynamics. And this is the model of the perceptron. So you have these multiple inputs coming into this single unit that summarizes things according to some sort of rule or conditional, and then there's an output. The weight loop perceptron, which is the first model that they use, follows the formal perceptron definition from section 3, which is the previous section. It consists of 21 species and 34 reactions. Apart from the core species, the weight loop perceptron requires a processed weight species here and fuel species E. So this is the weight loop perceptron computes the weight sum directly by transforming weights W into output species Y. The problem is that the weights encode the state of the perceptron, so their concentration must be preserved. Apart from Y species, the perceptron must also create backup copies of the weights. The perceptron can then restore its weights after the output production is over. And so this kind of works by preserving information and re-encoding it. So they give a table with all these different weights in it. Then the weight race perceptron is the second model. So the functioning of the weight loop perceptron is based on rather conservatively designed phases working in sequence. This approach works well since there's almost a one-to-one relation between the routines of a formal perceptron and those of a chemical perceptron. Nevertheless, the idea of a direct calculation of the weight sum and recovering the original state seems unnecessarily cumbersome for a chemical system. The weight race perceptron improves upon the weight loop perceptron by switching the chemical roles of inputs and weights. That is, instead of having inputs catalyzing a transformation of weights to a weight sum, which determines an output, weights simply catalyze the input to output reactions, as presented in figure 4, which is this figure here, which shows this process where in a binary mode function, the weights W catalyze or compete on the input-output reactions of X and Y. So X and W compete, and then there's an output Y. In the learning mode, which is B, the desired output species D0 and D1 transforms to weights W if the provided variant does not match the actual output. So in this case, your species transforms to the weights W if the variant doesn't match this actual output. So it has to match the output of Y. And this is this learning process where it tries to calibrate itself to the output. So that's this paper. The second paper is this feed-forward chemical neural network, an in silico chemical system that learns exclusive OR. So XOR is exclusive OR. This is usually used as a logic gate, but this is also Christoph Tucher is the third author on this paper. And this also has, a chemical neural network, so this is a similar type of system. Inspired by natural biochemicals that perform complex information processing within living cells, we design and simulate a chemically implemented feed-forward neural network, which learns by novel chemical reaction-based analog of backpropagation. Our network is implemented in a simulated chemical system where individual neurons are separated from each other by semi-permeable cell-like membranes. So this is where we have neurons that have these semi-permeable membranes, which is an interesting design feature because most neural network neurons don't worry about this too much. They worry about mathematical functions. So this is this is a compartmentalized modular design that allows a variety of network topologies to be constructed from the same building blocks. This brings us towards general purpose adaptive learning in chemico, which means that they want to be able to implement this in a chemical system, wet machine learning in an embodied dynamical system. So they talk about this as, sort of an embodied system. Having developed a family of individual chemical perceptrons, we wish to design a method for connecting these in a more computationally powerful network. So the network should be modular and allow for different topologies to be reconstructed. We achieve this goal with a feed-forward chemical neural network, or an FCNN, which is a network of cell-like compartments, each containing a chemical neuron as a module. Communication between nodes in the network is achieved by permeation through the walls of these compartments. So these compartments have walls. Walls are selectively permeable, and things pass between the walls, which allows the network's feed-forward and backpropagation mechanisms to function. So this is a little bit different than the way we usually think about neural networks, which is that they have connections that have weights, and then those weights determine whether the message is passed on to the next network or neuron in the network. Like standard single-layer perceptrons, each of our individual chemical perceptrons can learn the linearly separable binary two-input logic functions, such as AND or OR. So these are the simplest logic functions, AND or OR. It's that you have a logic gate that says, I'm going to, if such is true, there are two things that are activated in AND. In OR, if something is true, one thing or the other thing are activated. So there are different ways, different rules that we can use to, when a logic function is performed, to pass on information to the next unit. These simple logic functions, however, are incapable of learning the linearly inseparable functions XOR and XNOR, which is exclusive OR or exclusive NOR. So this is where you have exclusivity in some of these functions. So these are linearly inseparable. Here we demonstrate the FCNN learns each of these functions. To our knowledge, it is the first chemical system able to learn a linearly inseparable function. So this is a breakthrough kind of for these kind of networks, actually for neural networks in general, in that you have these linearly inseparable functions that can be performed in one step. And so this is, I don't know if there's a figure here, but this is the chemical reaction network. Again, this is, sort of a rehash of what we saw in the other paper. These cell-like compartments. So he brings up in this part, there's an interesting passage. Since Aristotle, Pasteur, more recently Maturana and Varela, cellular compartmentalization has been considered a central characteristic of living systems. This is a major motivation behind the modern study of protocells, which we talked about last week and a couple weeks ago as being something that people use to study minimal cells and the origins of life. And it's actually something they do in artificial life a lot. They work with protocells, which are these vesicles that you can load up with different things. You can boot them up with biochemicals or other proteins, and you can get them to do things, behave like minimal cells, just with enough, enough stuff in it to get it to do things. The analogy of the cell as a self-contained, self-sustaining regulatory machine is a familiar one. And the most basic requirement for such a machine is compartmentalization. So compartmentalization actually is interesting because it enables a lot of things. It enables local information processing. It enables, by extension, what we call modularity, or the specialization of functions in a certain place. It's bounded by something like a boundary or a membrane. And it allows for things, higher order things like redundancies and other things where you have multiple copies of something, so that if one thing is damaged or one thing goes offline, the other things can take over. And you can only get these sorts of properties with compartmentalization because you need to have specialization. You need to have individual units instead of having things as kind of as a generic system. And we don't see a lot of that in neural networks. You see some, like we see layers of neural networks, and we see some specialization of different layers. But people don't really talk too much about modularity. So this is an interesting way to approach this problem. And so they talk about a couple things here. They talk about their networks as modular and having these compartments. But that also implies that they have boundaries. And so the membrane boundaries are permeable. They have to be, and of course, actual cells are permeable. We have ion channels. We have gap junctions. We have other types of passageways in and out of the cell. And these things are specialized for different functions. So you can think about like an ion channel. It's very selective for certain ions to pass through, and it has a function. So this is something that is can be highly specialized for a certain thing that you want to do. And of course, in neural networks, we don't really talk too much about, we don't talk about like ion channels very much. When we model real neurons, we do. And this is kind of what they're trying to replicate in this work. So they model this mathematically, and you can see the math here. They talk about chemical neurons. The chemical neuron we chose as the building block for our FCNNs is the analog asymmetric signaling perceptron, or AASP. And these are two new variants of the AASP are used, one for hidden neurons and one for the output neuron. As a reference throughout the section, the rest of the article, Table 1 lists all the chemical species in the FCNN, so this is in the supplemental materials. This is an artificial life article, so it's at their artificial life journal website. So they go on. They talk. This is the table here with the inputs, outputs, weights, and the different chemical species that are associated with these things. Then they kind of go through these things: inputs, input weight integration, and they talk about basically the mathematical mapping of these things. So this is a reaction, a map of the reactions in the input weight integration step in a 2-input AASP. So you can see that the dotted arrows denote a catalyst. So this is a catalyst here, for example, or this one. And then this signifies a reaction in the network between x sub 1 and y. Each weight catalyzes a reaction, turning its associated input into y. So everything gets turned into y, while that input and y simultaneously annihilate each other at a different rate. So this is where you get these sort of dynamics, these kinetic dynamics, and you can classify it or you can characterize it in a network. So then they get to the learning part where they go through some of this. They have these AASP networks, and they show kind of how these work. Positive weight adaptation, negative weight adaptation, and an output comparison. So they're different types of learning that they propose. And then finally, we can talk about some of, what these are useful for, which is that you have these FCNNs, and they have a tree-like topology. So you can take a tree of these compartments, or you can take these compartments in say a cell or a structure, and you can map them to a tree. So you can show how these things sort of operate or maybe even evolve. And so this is a nice way of representing this. Okay, so thank you for listening to that. I hope that was useful in terms of interpreting some of our discussions that we've had.
Okay, go ahead, Bradley.
Yeah. So I just checked. I got a check-in from Alon Samuel, and he doesn't have anything to report, but I hear that there's an email thread between Dick and Alon and Thomas.
Yeah, I think Thomas has finally gotten my point. It's based on a very bad experience, which Susan might remember. I bought the first Apple camera. It was separate from the computer. Okay, so you plugged it into the computer, and we took axolotl eggs and dropped them in a tube. And after the initial transient, they should fall very smoothly. Okay, so I took movies of this, and the movies were completely jittery. The time interval between frames was very, very bad. Okay, so based on that, I don't trust digital cameras, digital movie cameras. Okay,
yeah.
And I think Thomas has finally got my point, and we'll try to do an experiment where the motion of something under the microscope slide that approximates the size of a diatom and does not undergo stop-go friction with the slide will move uniformly, and then check his camera.
The other thing is that beyond a certain limit in size, something that's about say a quarter of an axolotl egg, whatever that is, beyond that limit, the Stokes drop through water experiment doesn't work because they float rather than.
They float.
It's not.
The axolotl egg
equations for that don't work beyond.
If it floats, it's not going to move.
Pardon?
If it floats, it's not going to fall.
Yeah, float's not fall.
Yeah,
but
the axolotl eggs are more dense than water.
I can find you that exact limit if you want, because I wrote it up when I was in the drop experiments with the axolotl eggs that I was doing in the sucrose solution.
Yeah. To find the density. Yeah,
I did a density experiment where I had to
you know the experiment we did a long time ago? What was his name? The guy who was a high school teacher spent a year with us.
Yes, Flint.
Yeah. What? Russ? What was his name?
Flint.
Flint. What was his first name? Russ Flint.
Oh, okay.
Yeah, what we did is we hard-boiled an axolotl egg. Okay, you can do it just by cooking it a little bit. And then we sliced it vertically. And then we let the slices fall in a sucrose gradient. And they separated very nicely. So there's a gradient of density top to bottom, or bottom to top, highest at the bottom, inside an axolotl egg that's been hard-boiled.
And what I did was I kind of exploded the eggs. Well, not exploded. I put them through it looks like sort of a little mini guitar, tiny little thing that broke the eggs up into the sucrose solution, and I had some on the bottom, and then with the graduation in my sucrose gradient. And the pieces were just at the limit of the sucrose gradient. Were too small to be in that proper measure. But I figured they were just big enough to be over the limit.
Okay.
Size. And then axolotls do this interesting thing. They float on top of your sucrose gradient, like they just when the blastopore becomes its largest.
That's
actually
we did
sucrose gradient and become less and less dense.
Oh, okay.
And they fall again.
Okay, that's many cells. That's interesting.
That's the whole egg.
That might fit in with some of Steve Levin's ideas.
Well, I don't know. This is a known phenomenon. They when the blastocoel is finishing growing, they actually do this, and then they do that over and over inside, and then they fall. Then they become denser.
Interesting.
At this point, when they have risen, that's when I want to get out the optical coherence tomography and take an image of the blastocoel. I want to do this experiment, but like I said, I have to wait until I finished my other stuff for my PhD. But he said I could do that. He said I could do this at the end. I go, okay, it's a promise. This is the carrot.
Yeah, she's got an advisor who took her on and treats her like a 20-year-old.
Yeah, so he was surprised when I wrote a paper in the middle of a pandemic. I kept trying to tell him this is a family thing. Like, I have engineers in the family, and one of them was completely bored. And so I told her to design me mounts for my microscopes, and she did. So I got someone to 3D print it, and my husband put it together because I don't have that kind of patience some days. and then my brother-in-law designed some of the electronics for it because that's what he does. And I put them all in the acknowledgement, and my daughter is the co-author, and she wrote part of it. So it's like, what did I do? I wrote some intro and history of this, and then I handed it in. I don't know. It's like,
okay.
Anyway, so yeah, it was a family paper.
Yeah, something I wanted to bring up.
Bradley, if you could try to get Steve Levin to give us a talk.
Yeah, we were talking about that earlier before you came into the meeting.
We had an informal meeting with him yesterday.
Yeah.
And he does have some good ideas. He's 90 now.
Yeah.
But his brain is just fine.
Yeah.
Okay.
Yeah.
We're twisting his arm to write a book that he's been promising for at least a decade.
He doesn't want to do
And that would be the time to do it, yeah.
You should try it as a picture book first and go take photos of all his.
Yeah, I think the thing to do, Susan, is draft the text and let him tear it up.
Oh, well, I could start it as a paper because I have to do it anyway for my project.
I started a paper that turned out an almost 2,000-page book. So.
Oh, okay. Oh no, that's where I would start, is like with this project that I'm doing, and I'll definitely send it to him, and maybe he'll expand on it. That would be a good start.
Okay. But I mean, if he, I think his ideas are good. So despite his.
Okay, excuse me, sorry.
Yeah, so I wanted to revisit something I talked about last week, which was the. I went through that book and I was looking at how you might simulate tensegrity networks. And so I'm going to share my screen.
Thank you.
I wanted to bring up
yesterday especially is that he doesn't use elastics for the string component between the rods. He uses fish line.
Oh, okay.
Which is very rigid. Okay, and he thinks that's an essential part of it.
I think so.
Huh.
Yeah, he may be right.
Yeah.
At any rate, he's claiming that as a result of this, if you squeeze one part, the forces are distributed over the whole structure, which is interesting. So we're looking for some simulations of this where you replace one string by an elastic, so you weaken it, and see if the weakening is distributed over a whole structure.
Yeah. Yeah.
Okay.
Yeah. So squeezing one part would be like, what would that entail?
Well, in his case, what he's saying, he said two things. One is, if you try to lift a heavy weight,
right,
the pressure in your eyeballs goes up.
Yeah.
He's taking that as evidence that the whole body is an integral tensegrity structure which distributes the forces.
Okay.
And another example he gave is the heel, he says, is very soft as a bone, but if you're running, it hardens while you run.
Okay.
Okay, so the rate at which the forces are applied to it alter its stiffness. Okay, so these are new concepts to me. So
yeah,
we're trying to get him to get these ideas out because nobody else has written about them.
Yeah, yeah. It's hard to find references on different things, especially about networks. There isn't a lot. Yeah. So
he regards the body as basically a fractal structure. Whoops. Oh, okay. The body is a fractal structure of tensegrity structures.
Okay.
So there's your larger ones, which contain smaller ones, which contain smaller ones, etc. And so the whole thing reacts. The whole structure reacts. It's an interesting idea, but it's testable.
Yeah, yeah.
There is a paper that was recently written about microfilaments being a tensegrity structure. Now I'm going to turn off my camera. Things are going too slow. Okay?
Yeah.
Okay. See if that helps.
Yeah, yeah. Okay. So
Are slow.
Yeah. So this is the book that Susan sent to me, and this is let's see if it comes up. Computational Modeling of Tensegrity Structures by Gann. And this is where I got a lot of the MATLAB code for this example.
Oh, okay.
So in the book, I think it's like chapter 2, they lay out MATLAB code for simulating a tensegrity network. And it's like basically you define the network, you define the nodes, you define the edges, you basically build a matrix, and then that matrix is the way they have it set up. It's like -1, 0, and 1. Those are the three states. And then like it's either there's neutral tension, there's positive tension, or negative tension. They define that in the book as to what those mean.
They don't use, what do you call them, rods and strings?
Well, it's basically the same thing. So the rods would be the edges, the sort of the nodes. I guess the rods would be the nodes. The rods would be the nodes, and then the strings would be the edges. So it's just a very generic structure.
What of the -1, 0, and 1, which corresponds?
Well, that's actually, so what it is like a connectivity matrix.
Oh, these are the nodes. It's a matrix of the nodes.
Yeah.
Okay.
So it's like you have, I don't know what the notation they use.
And do they assume linear springs between the nodes that have what shall we call it, the elastic component?
Yeah. I think they assume linearity. But the way they set the problem up,
Steve was arguing against that. He said that's a mistake. They should be not highly nonlinear.
Okay. So basically, they have it like this. You have this I sub ij, which is a connectivity matrix, where you have like I and j, and then you have you have three states: +1, 0, and -1. And you can make different assumptions about the connectivity, like whether the connections are linear or not. But you end up with these three states of force, and then those states of force are in the matrix, like this.
Okay, so the matrix is independent of the mechanics of the sides.
Yeah. This is just characterizing the mechanics.
Okay.
Yeah, and so that's the way they set it up, and then you can do different matrix operations. In the book, they talk about doing like support vector machines and finding eigenvalues and things like different matrix algebra things that you can do with these matrices.
Did they do random matrices?
They didn't do random matrices in the book, but you can easily do those. So,
you could, okay.
Yeah, so this is the code here. Let me bring it up. This is like in MATLAB, they call this an M file, which is a function. I don't have it set up as a function here, but I have the code. So you have 3 dimensions. These are your inputs, X, Y, Z, and then this is your matrix C. So this is these are the values of these states. So this is, I guess, your set of, your 3 things, and then this actually, I think these are your spatial coordinates of the nodes, and then these are the force. This is the force these are the force vectors as a matrix, and then this is, and then you work through this. You do some matrix algebra to get to down to the bottom here.
Okay.
Yeah, and then you have to find, the way they have the code here is you find like the rank for different things. So you have the rank size, and then so there are different ways different things you can calculate out of the matrix.
Okay, stupid question. Go up to the top again.
All right.
If you assign the wrong coordinates to X, Y, and Z, okay, would it settle down to these coordinates?
I don't know.
You don't know.
Yeah.
That's the question of squishing it.
Okay.
Well, I have that finite element analysis paper that I maybe should send you, or maybe did I? I don't know if I did.
I don't think so.
Did I ever send you? What's its name? Did I ever send you equivalent mechanical properties of tensegrity truss structures with self-stress? Did I ever send you that?
You may have. I think I have some papers on it.
It's okay.
Yeah.
Okay, Bradley, where do those numbers come from for the X, Y, and Z?
They just generated them using the example. So they have a set of.
Are they empirically?
The ones in the book, I don't think they are. I think they generated a bunch of values to demonstrate what it can do. But you can generate your own values from it, and it works.
Yeah, because obviously, if you generate the wrong values, it's going to move.
Right.
That's why I asked, does it settle down to those values, or where do they get them from?
Oh, I think it's just like they build like a standard tensegrity structure, and then they just put in numbers. But like like a physical toy model like Susan has. But the idea would be that you would be able to characterize any structure like that and evaluate it.
Okay, but where's the program that generates the XYZ values?
There's no program for that. For that.
I couldn't make those out.
Yeah, there's no program
I couldn't make those numbers out of my head.
Oh, yeah, I mean, it's just like something that they've like made from an example that they use, but
okay, fine. But obviously, in the general case, you need to generate them.
Right. You need to generate them, and then you have to figure out like what the how it behaves physically. Well, I guess that's something to be done in like a physics simulator, like COMSOL or something, where you can actually evaluate if something is in a position in the three-dimensional space.
Yeah.
Or how does it behave?
Yeah, the net vector, obviously, if the sum of the forces at every node has to be zero when it settles down.
Right.
Okay.
Yeah.
Anyways, this paper that I put the title of it in the chat, and I can send it to you too.
Oh yeah.
And I have another paper about material for wearable electronics that has a J curve, like Stephen Levin says that all tensegrity structures have, and they've taken a close-up look at this material, and it looks sort of tensegrity-like.
Okay.
So I have that paper too. So maybe I should send you those two papers.
Yeah, I think I have the first one, but not the second one.
Oh, you have the equivalent.
Yeah. Yeah, so I think I have it in this folder, and I'll talk about it in a minute, actually.
Okay.
Yeah, so this one, I wanted to finish off the discussion here. Yeah. Oh, that's the wrong one.
Oh, Bradley, another stupid question.
Yeah.
Do they discuss waves in tensegrity structures?
No, they don't.
They don't. Okay, because those could also be important.
Yeah.
I think there was a paper, I was looking through it, that had something about waves.
Okay.
Measuring them, using waves to get okay, I'm losing it. The real and imaginary parts of a tensegrity structure, like the
Yeah.
G and G prime and G prime prime. G, whatever.
Yeah, again, maybe you could study that here if you distorted the structure by changing the XYZ.
Right.
Yeah.
Then maybe if you followed individual coordinates, they would oscillate and show you that the wave is going through them while it settles down.
Yeah, well, I'm hoping to put a wave through the tensegrity structure so I can get.
Okay. It's interesting to know if they act like damp waves or not.
Yeah.
You know what dampens the waves is if you put water into the model.
Provide a friction, yeah.
Yeah, it does. I have a paper about that too. where they have a model and a truss model, and then they put water in it, and it dampens the oscillations. So people have been fooling around with this with models for a while. I should just, if you want any of these papers.
Yeah, that's why. Is there a decent book on that does it?
That is the only book, as far as I know. But yeah, there needs to be a decent book. That's why Stephen Levin should have written it already.
Yeah, well, okay. I think you're going to end up having to write it and have him critique it and fix it.
G is like elasticity, except that it's got a real and imaginary part to it.
Okay, thank you. All right. Bye.
Okay.
Yeah. So this is the code. It's run in SciLab. So SciLab is sort of like MATLAB, except it's open source. You can run a lot of MATLAB code in it. And the thing about SciLab and MATLAB is you can do matrix transformations in it easily. So this is just the code that's run here. You have this matrix, identity matrix, or well, it's actually not an identity matrix. It's a matrix of the
memory.
states.
There's a language called Octave. Is that also a
yeah,
type?
I think it's very similar. It's very similar. And then this is where you get the rank for the different components of the matrix, and you get a graph like this. This isn't very informative. It just gives you like a rank of for the different dimensions. Does rank have
anything to do with whether it collapses or not?
I think if the rank is above a certain value, it gives you, like, basically what they're trying to do with this code is evaluate the state of the structure, if it's stable or not. And so I think at certain levels of like if the rank is above a certain number, it's stable, and if it's below a certain number, it's not. But this isn't a very good way to evaluate it.
So we could use that as an analog and say, if it's stable, it's alive, and if it's not stable, it's dead.
Yeah, basically. That's what I got out of it, and I think that would be a nice heuristic. But I think there's probably a better way to figure out what a summary statistic for the structure. That's basically what this is. But yeah, so that's all. Yeah, so that's one option for computing this stuff, and it's there probably maybe better options. But or things could be added to this as we would want.
Susan, I got a stupid question for Susan. Has anyone done a tensegrity analysis of cell division?
Not that I know of. There's somebody who is looking at a tensegrity system dynamics. Anyway, has anybody done? Say that again.
Has anyone done? Yeah, you take a sphere, divides it into two spheres.
Oh yeah. Have they done that? No, not that I know of.
Not that you know of. Okay, that's a good problem for the future.
Yes, dividing it up or taking
minimal requirements for cell division.
Yeah.
Okay, I'm trying to find some of these references I've been talking about for you.
Okay.
It would be interesting to, Bradley.
Yeah.
If you go back to the nematode, we know that most of the cell divisions are asymmetric.
Mm-hmm.
And it could be that one could make a tensegrity model that is trivially asymmetric in its cell division.
Yeah.
Okay. So some of these things may not be set up by the cells, or you can almost. You made toy balloons.
Yeah. You mean like balloon animals, or?
Yeah, balloon animals. You ever see how hard it is to get them symmetric?
Yeah, because they have given certain places. Yeah.
Yeah. They tend when you try to squeeze them into two parts, they have different sizes.
Yeah.
Okay, so that might be a tensegrity phenomenon. I don't know.
Yeah. Yeah, that's. So this is the paper Susan was talking about, I think. This is truss structures. Yeah, so this is, make it a little bit bigger.
Okay, remind me, what's a truss?
A truss is like a building, a high-rise building, and you're just looking at, you know what? Can I share my screen?
Yeah. Yeah, let me,
let's see. I'll just share my screen. There's the share screen on the thing.
down at the bottom and the third one from the left. Third button.
Yeah. Hey, thank you. Okay. So there's the truss that's being pushed around. There. You see it?
Okay, the hard parts.
There's like dowels and strings in this. This is a tensegrity structure.
Yeah.
And a truss is just has dowels and strings. It's not necessarily a tensegrity. Like you can bolt the hard parts together and then have strings hold up the sides, like a flagpole. And that's a truss structure.
There's a wave going through it.
Yeah, and put a wave through it, and then they check to see which system, see if both of these different types of modeling matched. And they sort of did, like the TSG, FEM, and ANIS.
Oh, okay.
And I'm not using ANIS, I'm using COMSOL. But ANIS and COMSOL are, well, similar.
Okay.
It's just that COMSOL is multi-physics. So you can use, I could put light through it. I could simulate putting light through the structure. And that's why I chose it. But I almost want to use ANIS because Dr. Zhang knows it. And I get hung up with the darndest things in COMSOL.
Yeah.
Anyway, sorry. There's the one with the water in it. I just saw that there. See that?
Yeah.
Okay. Yeah.
Here, and then this is fluid immersion and what happens to the oscillations.
Okay.
And this is a tensegrity structure in fluid.
So this is a good example of the XYZ coordinates, like where the node is in space,
right.
And then it's experiencing shifts in position, or for the different nodes, the one on the right.
Yeah. Well, you can't hardly see the oscillations on the left side without the water, and then or the fluid, and then the right side has with fluid, and you can see the oscillations in the different directions. And they're just looking at one node on the tensegrity structure. And
okay.
Anyway, I gave you the reference to the paper so you can find out what on earth node they're talking about.
Yeah. Okay.
Yourself, yeah, because I forget. Like this was a while ago that I presented this. And of course, then
they assign a mass to the edges or to the nodes?
It looks like they're tracking nodes.
No, do they assign mass to them?
Oh, mass. A truss structure, if they're doing a truss structure, maybe it doesn't have mass, which is why I was complaining about, you want me to find the center of mass of a truss structure in COMSOL. I don't think that works.
Right, unless you assign weights to
yeah.
assign mass to the components, then you can do it. Okay. Yeah, this is looking like a proper book taking into account these things and extending them. And incorporating Steve's ideas is probably the right thing to do.
Yeah. And this is a T1 transition, and I didn't ask him about it. He says they don't move, but they reconfigure themselves by changing shape.
Right.
Oh.
Yeah, did you read Wayne Broadland's paper on that?
Partly.
Oh, okay, you got it. You got it, okay.
I've got the paper, yeah.
Oh, okay, good.
And then cells also do this. They extrude. Anyway.
Oh, yes.
They'll do that. No, we don't want you in here.
Look, they also...
Apoptosis.
Yeah, I think there's an opposite of extrusion. If a cell undergoes a cell division, I recall empirically the cell goes down and then divides and comes up again. That's two cells.
It goes down?
Yeah. That's my memory of it.
Okay, well, that would be this cell division portion here.
Yeah.
I was kind of interested in this T2 transition.
Mm-hmm.
But, well, I'll get there.
Okay.
They said they just want me to do this top regular one and measure it. And I'm going, fine, but it's really easy to do some of these other things. And
yeah,
they didn't seem interested in doing that. But maybe I could just do it on the side because I know it's important. And then they'll all of a sudden discover that it is. And I'll say, oh, well, here's the solution.
Yeah, well, what's important to an engineer is different from what's important to a biologist.
True, and I'm going with the engineers on this. We really just want something that mimics part of the behavior of the cells. Like to build a material that covers part of the J curve that he's talking about, or stress strain, the stress strain curve. If it covers part of it, that's good. I'll get another material for the other part of the curve and a third one for the top part or something. So then I can cover the whole curve, stress strain curve, and then we can improve optical elastography. That's talking, not but if I had my way, that's what we would be aiming for. Because trying to get this tensegrity structure to be a perfect mimic of cells is like asking for, you want me to do what, where? It's not going to work. According to Stephen Lepp, it definitely not. Because it has to be a fractal structure with tensegrity structures making up the rods and strings, and then the rods and strings have their own tensegrity structure. So how do you do this? Well, the engineers are not going to be very pleased because they want to do inverse problems, and it has to be linear.
How long do you want to start your postdoc work before you doctorate?
Sure.
Okay.
Hi, Jesse.
Bradley, can I switch topics?
Yeah.
Yeah, did you get that paper I sent last night from this guy named Lace on articulated structures and basilaria?
Yeah, I saw it. I didn't go through it, though.
Oh, yeah, it's a very difficult paper for me.
Yeah.
I was impressed that we tried to get a simple version by inviting him to speak.
Okay. That would be good.
Okay.
Yeah.
There's a lot of it's all mathematics.
Yeah.
And a lot of so getting the basic ideas out of it would be best by questioning him.
Yeah. Yeah, that would be good. I'll take a look at it after the meeting and see.
Yeah, he asked me for an evaluation of the paper. I deferred to Thomas.
Okay, yeah. All right. All right. Yeah. All right. So yeah, let's.
Okay, cool.
We have the. Well, Susan shared that paper. Let's see if I can share my screen here. Okay. So this is the paper she shared. Equivalent mechanical properties of tensegrity truss structures with self-stress included. So this is the. They talk. Let's see if there are any figures in this paper. Well, these are the matrix. Well, these are the block matrices here, where they define mechanical characteristics can be determined from the inverse elasticity matrix, which is H, and then these are the components of H, and then they take blocks out of that matrix.
There's your G there's G. You see, you have E, and then you have G.
Yeah, on the right hand side of the matrix.
Yeah.
So what is G again? Is
E is elasticity.
Okay.
And G is similar, except it's got a complicated component, imaginary component. It's something you find through oscillating the material.
Okay.
All right. I was trying to find its name, which has escaped me, which is kind of dumb. You know, I'm an electrical engineer, right? Not a mechanical engineer. I was just like, wow.
There is no room for disciplinary bias.
Yeah.
Yeah, so it's quite obvious.
Yeah. Yeah, a lot of this stuff, like if you're simulating, it gets pretty deep into matrix algebra and matrix manipulation. So the mechanical properties of 5 typical tensegrity modules inscribed in the cube of edge length A are determined and evaluated using the proposed continuum model. So they have things like 4-strut simplexes, 3-strut simplexes, an octahedron, a tetrahedron, and an X module. So they use a series of different structures to do this. And then they analyze structures consisting of cables and struts again. So these are not biological structures, they're just generic structures that form this network. Then they have this parameter K, which is the EA cable over EA strut, where E is the Young's modulus, which is defined by and then A is the cross section, and then the self-equilibrated system of the forces is represented by this parameter S over EA, where S is a free multiplier, and then EA is that quantity there. So these are the matrices that they derive from this. And they do the calculations, and then this is what they look like, these 4-strut simplex modules. So they have this structure here, and again they have this XYZ, these coordinates. They have the forces acting upon the nodes, and then those are transmitted throughout the structure, and then you do the calculations and you can figure out whether it's stable or not.
I like the stability analysis in this. That's something that Dr. Sharif, who's the electrical engineer in the party, he was very interested in where the thing was stable and not stable. And G is the shear modulus.
Okay, shear modulus,
Yeah.
Yeah. But that's that paper.
I like.
Go ahead.
I would like this idea.
Yeah. Then I think you sent me 2 other ones. There was this paper on Turing and wave instabilities in hyperbolic reaction-diffusion systems. This is I don't know if I did maybe I added these in. And learning the stress-strain fields in digital composites using Fourier neural operator. So there are a couple of areas,
I think I sent you that one because, or if I did, it's because I'm going, it's what?
Yeah.
Yeah.
Not something Fourier would recognize.
Yeah. Yeah, these are
Don't know.
basically they're analyzing materials for their sort of the properties and performance using these neural operators, which this is a Fourier neural operator. I don't know what a neural operator. I've never heard of a neural operator actually, but I think it's like some sort of Oh, okay. So it has. I think it's like some sort of neural network that they're
It's
using.
using a neural net for the solver.
Oh,
Okay.
Yeah.
Instead of a matricy? Is it?
Yeah, instead of using the like finite element solver or something like that.
Oh, okay. All right. So instead of finite elements, it's using a neural net.
Yeah. Yeah. And yeah, they plug it. So they do. This is sort of high-end machine learning. So you have to have, and I guess you have to have a lot of computational power for typical.
Well, they turn out to be very, very efficient as well. I mean, it's a cool property of them. So, like in astrophysics, they're doing big like galaxy computations, and they're finding that the neural nets can do these orders of magnitude more efficiently.
Oh, okay.
Oh,
wow.
Maybe I want to do that with my simulations, just because the department I'm with would love that.
Oh yeah.
Yeah, this is a really hot area of current like deep learning to solve ODEs, PDEs with these. Sorry,
You're calling it what?
Well, so some of these are pins. Sorry, my daughter's playing with a machine here. Physics inspired neural nets, and other these neural operators.
Yeah. So they're learning the mappings between finite dimensional Euclidean spaces, and they're predicting these outputs. Or they're solving equations basically. And then there's a Fourier neural operator which does a similar thing. It's doing, I guess, a Fourier analysis. Yeah. So this is, and then this shows you kind of the diagram of the system where you have an input, you input it into this neural operator, and you go to output. So that's something that's an option too. And then of course it relates, the third paper here, that of course some of this relates to pattern formation and some of these observations of wave instabilities. So these are reaction-diffusion equations, hyperbolic reaction-diffusion equations. And this just talks about some of the diffusive instabilities. And it doesn't really talk about tensegrity, but it's kind of talks about some of these things about pattern formation. So that's all I have on that.
What is a hyperbolic diffusion?
It's just, I guess, a type of equation that they use to model reaction diffusion. So this is a hyperbolic reaction diffusion system. So this is standard reaction diffusion systems are best approximations of the mean field behavior of particles. So an example is the infinite propagation speed of disturbances in these parabolic systems. So they're looking at like reaction diffusion systems, but they're looking at disturbances within them as they operate. So one phenomenological way of addressing the issue with propagation speed is to consider a hyperbolic reaction diffusion system, which can be shown to admit both finite speeds of propagation and behavior compatible with the standard case and the singular limit, at least for dissipative dynamics. So this is hardcore dynamical systems, looking at reaction diffusion as a process, but then looking at how it can be perturbed or show instabilities in it. Yeah.
Okay, I'll have a look at that. Did I send that one to you? Or
I'm not sure. I can share the papers though. Reshare them.
Yeah. Okay.
Yeah.
All right. I just I need to know about all of kind of the broad breadth of how to solve for these things, or I'll get myself into trouble when being examined. Yeah. And going, no, I didn't want to use that system because of blah.
Yeah.
Yeah.
Oh, that's great.
Yeah, it helps. Okay.
Yeah.
Besides, my niece might know something about some of the neural nets because she was looking at quasars and nebula. That's it. She was trying to figure out the shape of a nebula from just one direction. So you only have image of a nebula from one direction because that's where we are in the universe. And she was trying to figure out their 3D shape from just looking at it from one direction.
Okay.
Oh boy.
That was her PhD thesis.
Oh, okay.
She just attempted it.
Susan, is that electronic?
Well, it's all neural net.
No, it's the thesis. Can you send me the thesis?
Oh, okay.
Yeah, it's quite interesting.
Yeah, you'd like to see it too, Bradley?
Yeah.
Okay, send thesis. Make myself a note. All right. And I think she's got it published by now.
Okay.
Okay.
Well, that was great. Yeah. I think there's some things to follow up on here. And, I guess we'll continue the discussion. This is a pretty interesting area. I'm glad we've kind of gotten back into the tensegrity area. Yeah. All right.
Okay, great.
All right. Thanks.
Okay, thank you for this discussion. It helps. Like all of this really, really helps me.
Okay.
Right.
That put the pressure on, but we got to turn that paper in by December 31st.
The one that, I'm working on that. I'm still, I've been working on it over the weekend too. So
yeah, with the vehicles and perhaps the tensegrity structure.
Yeah.
Okay.
All right. Sounds good. All right.
Okay.
Have a good week.
Bye.
Bye.
You too.
Okay, bye.
Bye.